Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is meant for 2nd September 2020, and we have been discussing about the new baby of biology, that's biotechnology, and I said that this has got a lot of future and this is going to be the flag for all of us. It saves a lot of money, lot of lives in future with its technology. So basically we said biotechnology is biology with a combination of technology. So we are using the technology in making the DNA corrections, in making the gene corrections, and finally in making even a recombinant DNA or hybrid DNA. So we were talking about various steps involved in making our DNA, recombinant DNA. Now in this session we are going to discuss about what are the various tools required for making our DNA technology. As I said, our DNA technology, recombinant. First we'll see combination of DNA. All of you have already studied in our previous chapter, molecular biology, what is the combination of DNA. We said basically DNA is a polynucleotidal chain. And when I say polynucleotidal, many nucleotides are present in DNA, correct? What is a nucleotide? Nucleotide is a combination of three chemical molecules. One is sugar molecule, deoxyribose. Second one is phosphate molecule. And the third one is nitrogenous bases. And in that nitrogenous bases, we talked that there are two types of nitrogen bases, double ringed ones and single ringed ones, purines and pyrimidines. And in that purines and pyrimidines, we talked about basically four nitrogenous bases in DNA. That is AT, combination always, AT, and GC, guanine and cytosine. So adenine thymine combination, guanine cytosine combination. So that is the combination of DNA. And what is recombinant? We are adding something else, something new from other source. If we add to the DNA of a particular organism, that becomes a recombinant DNA. So what is that we require to do that? First of all, we require restriction enzymes. So we say restriction enzymes are of two types, restriction endonucleases and restriction exonucleases. So first and foremost, we require this tool, restriction enzymes. Second, polymerase enzymes. Third, ligase. Ligase again is an enzyme that ligases, adds bits and pieces, vectors, somebody to transfer the rDNA, prepared rDNA into a suitable host. So we need these 5 tools for making recombinant DNA technology. So first one we discussed, first tool, restriction enzymes. These are the enzymes which are used to make cut in DNA. That is the main purpose. What is the purpose of a restriction enzyme? To make a cut in DNA. And once we make a cut in DNA, of course, as you are seeing in the picture, it's not a scissor or a scissor that can with which we can cut. Here it is DNA. Where is DNA? All of you know. First of all, a cell. Okay, first an organism. From organism we are entering into systems. From systems we are going to organs. From organs to tissues. From tissues to cell. From cell to nucleus, and inside the nucleus chromosome, inside the chromosome DNA. So that is to be cut. You can imagine how much it would be difficult to cut a DNA which is present inside the chromosome of a nucleus of a cell of an organism. That is the interesting thing. And in 1963, 2 enzymes were isolated from E. coli, Escherichia coli, which restricts the growth of bacteriophage. One enzyme added methyl group to DNA and other cut DNA, and the enzyme that cut the DNA is restriction endonuclease. Endo means inside. Function of restriction endonuclease depends on specific DNA nucleotide sequence. It is not that the restriction endonuclease can cut DNA wherever it likes. It's not so. in a particular nucleotide sequence should be there, then only it will cut. And it can cut at a particular point, or it can cut at the ends, either the beginning or the last. So it is not that it can cut anywhere. So first endonuclease isolated was Hind2, and studies proved that Hind2 always cut DNA at a particular point by recognizing, this is important, specific sequence of 6 base pairs, and these are called recognition sequence of enzyme. So 900 restriction enzymes are already isolated from 230 strains of bacteria. This is important. 900. So a lot of research is going on, though I said it's the baby of the biology. And then restriction enzymes belongs to nuclease class of enzymes. So there are, just now we said there are 2 types. What are those? Exonucleases and endonucleases. As the name suggests, exo, outside, removes nucleotide from end of DNA. On the sides, one of the sides, it can remove the nucleotides. Endo means inside. It cuts at specific positions within DNA. This is the difference between exonuclease and endonuclease. And how do we name this? While naming restriction enzymes, first letter represents genus name. Like for example, EcoR1. When we say first letter, what is first letter? E. E represents genera, genus name, Escherichia. And the next 2 letters, CO. CO represents coli, specific name, species name. So genus name and specific species name. Eco represents Escherichia coli. And 4th letter represents R. R means rough strain. You know, in every bacterium or prokaryote, there are 2 strains, rough strain and smooth strain. So some granulates will be there on the body, then we say rough. If it's not there, it is smooth. So R represents here rough. And what about this? EcoR1. 1 is the sequence or order in which the enzyme was isolated from the strain of E. coli. This is how we name restriction enzymes. So EcoR1 stands for Escherichia coli, rough strain, and of course the sequence. Like if it is 1, it is the first strain, that is first enzyme that is isolated. If it is RY13, it is a 13th strain. That is the sequence. The number represents the sequence, right? Now we go to the action part of it. What these restriction endonucleases do? As we said, they cut at a specific recognition site to bind and cut specifically each of the 2 strands of the double helix at their sugar phosphate backbone. And this restriction endonucleases recognizes a specific palindrome nucleotide sequences. What is this palindrome? Now palindrome is something that has got the base pairs in both the ways. When we read, it reads the same. Like for example, here he has given example. Before I go for this, I take out of biology one example which you are all familiar. Take for example Gadag, G-A-D-A-G. So when you read from left to right, it is the spelling is same, G-A-D-A-G. And now you read from right to left, again it is G-A-D-A-G, Gadag. So such a structure is called palindrome. Similarly, I think you can quote many words like Malayalam and in Kannada, Vikatakavi, like that. If you spell on both the ways, left to right and left to right, right to left, then it is called palindrome. So in DNA, what is palindrome? Look at that. The direction is 5' to 3'. So 5', 3'. And what are the nitrogen bases that are arranged in sequence? G, A, T, T, C, right? And you read this from this end, 5' here to 3'. So always reading is 53. When you read in this direction, it is G, there also G, A, T, T, C. So when you read on both the ends, the sequence is same. That's palindrome. So palindrome in a DNA is the sequence of base pairs that reads the same. You must remember this, that reads the same on two strands when orientation of reading is kept in the same direction. So normally this palindrome is asked, what is a palindrome? Give an example in your CBSE exam, 1 mark question or 2 marks question with example. So keep remembering what is a palindrome. Now restriction endonucleases cut the strand away from center. Look at this, it's cutting from away from palindrome sides, but between same two bases on the opposite strand, leading to formation of single stranded portion at the ends. They are called sticky ends. And what is sticky end? Look at this. This is the sticky ends. Here you can see this is the sticky end and this is the sticky end. Already we have discussed there is a weak hydrogen bond pairing this nitrogen bases. And I said sometimes it is better to have some weakness because that weakness becomes our strength. So here the weakness in DNA is hydrogen bonding, and that is becoming strength for us because we can break that weak hydrogen bond and make new DNA structures or hybrid DNAs or recombinant DNAs. And who is doing this? This restriction endonuclease is cutting it. The DNA, this is normal DNA, and you can see the restriction endonuclease is cutting at a specific point, and then once it cuts, then again the two counterparts are to be joined. Who is doing the duty of joining this? There is another enzyme called DNA ligase. So restriction endonucleases cuts at a specific point and DNA ligase joins it, thereby making the restriction new rDNA technology. So we have got an enzyme like endonuclease EcoR1, and it is source from where we obtain this Escherichia coli bacterium, and restriction recognition site in double strand DNA is this. You can see the arrow mark at G, it is cutting. So once the restriction and EcoR1 cuts this, what happens? The top portion G gets separated and rest of the nuclear sequence is there. Similarly at the down, the G gets separated here and the rest of it is present here. So we got 5' overhang. Similarly PSTL, this is Providentia stutai. So here also at specific site there is a cut at guanine, and you are seeing here 3'. And similarly SMA1 or Serrantia marcensis, again specific site here you can see the cutting edge here, so that the DNA is cut into blunt ends. So you got a similarly HAE3 Haemophilus aegyptus, and here the endonuclease is cutting at a specific site here, making the broken parts of the DNA. Similarly HPA2, here at the site of C, cytosine it is cutting, and here also you can see cytosine. So we are having the broken parts. So the purpose of endonuclease, restriction endonuclease, is to cut the DNA at a specific point. And where is that this is applied? Application of restriction endonuclease. It is used in the field of genetic engineering to form recombinant molecules of DNA. The purpose is to form rDNA. So this composition of DNA from different sources. Same restriction endonuclease, vector or source DNA, same kind of sticky ends which can be joined by DNA ligase, just now we said. So here take a paper and draw this. The enzyme cuts, now there are two DNAs, one is vector DNA and another is foreign DNA. This is we are selecting it, right? Now when we apply this EcoR1, it cuts at specific sites we said. Now you can see here the cutting portion here, and here also cut portion EcoR1. Once the cut portion happens, this is what the vector DNA forms, and this is what the foreign DNA gets formed. And now the DNA fragments join, this joining of the fragments will take place because of DNA ligase. Now you can see how new combination DNA, we call it recombinant DNA, is formed. You are seeing here same GC will be there, here G, here C will be there, and here also CG will be there in this portion, here CG, and the combination of AATT So here you got AATT from the sticky end of the foreign DNA, and the corresponding base pairing TTAC. So this is a T, this is a TAC will be formed, and this is how rDNA is formed. Now we can also represent this diagrammatically. How? This is how. So again, take a paper and draw. This is foreign DNA. We are extracting it from different sources, and this is vector DNA, plasmid. What is a plasmid? Extra chromosomal circular DNA, we said. Remember, plasmid is a extra chromosomal circular DNA. So you are having a circle. Now when we apply EcoR1, it cuts at specific points. See, cut, cut, cut. So it is cut. Now the ligase join foreign DNA to this. Now one of the pieces, now like I am picking up this, is joined here. So when it joins here, you get a rDNA. Now what is that we are doing? This piece is having a desirable gene, what we wanted. For example, we don't want some genetic disease to happen to human beings, and this gene is responsible to cure that genetic disease. So what we do? A gene of interest, we say. This gene of interest is incorporated here, and once we get this rDNA, recombinant DNA with the gene of interest, we transform this into E. coli, because E. coli can be cultured naturally and it is present in our small intestine. Now once it is transformed into the E. coli, we allow the E. coli to divide, and bacteria will undergo binary fission, multiple fission. As they undergo many fissions, how many fissions the E. coli undergoes, that many rDNAs are formed, and then we extract these DNAs, recombinant DNAs. So that since that recombinant DNA is having a gene of interest which cures, let's say, some genetic diseases, or which prevents certain diseases without any tablet, without any injection, we are successful in this. So this is the crux of biotechnology. Keep on practicing the